Hello, everybody. It's a real pleasure to welcome you to this College of Science Connects seminar. I am just delighted today to welcome Neel Joshi, one of our star junior faculty members, who will describe his research to you, but I'm not going to go there because Professor Penny Buning, who is head of our chemistry and chemical biology department, will do the honors to introduce Neel. What I want to do, though, is to give you a little context of how we view the College of Science at Northeastern. I moved to Northeastern 6 months ago. It's really flown by, but 6 months ago to become Dean of the College of Science. And I am absolutely thrilled to be here. This is a fantastic university. It is fantastic in its ability to think about the future of education in the most innovative and can-do way. And that is incredibly powerful. In the College of Science, we have the theme Connects. Connects means that we connect our top quality research with outstanding education of students at all levels and with innovation and entrepreneurship. Research lies at the heart of much of what we do. It informs our innovation, it informs our education, and it is the way that we are tackling the great challenges of our planet. The seminar series that you are participating in is a way to showcase some of the outstanding research that we do. We enormously value your interest. We enormously value your support of our research and other programs. Research is for everyone. I say that every undergraduate should spend some time doing research in a wet or dry lab research group, thinking about what are important problems, how you tackle them, how you set up hypotheses, how you refute or support hypotheses. This is how research goes forward. This kind of research, this approach to research of thinking deeply about problems and how to solve them, is how we are addressing the COVID crisis. Work done in millions of hours of research work, some of it done at Northeastern, is what is helping us solve COVID. It is scientific research that is the way that we address the great challenges across our planet and even into the cosmos. Chemistry, chemical biology is one of the enormous fields. Molecules are what have built the chemical industry, what have built the medication industry, and what drive us forward in a myriad ways. I am, as a chemistry major, as an undergraduate, I am completely sold on the importance of chemistry. It permeates everything that we do. Almost every field of science is connected with chemistry and with chemical biology. So for me, today, it is an enormous pleasure to have Neel as the representative of our research in the College of Science. And it is an enormous pleasure for me to introduce Professor Penny Buning, who heads our chemistry and chemical biology unit and who will take over. Thank you so much, Penny.
Thank you, Dean Sive. So it's my pleasure to introduce Neel Joshi, who is an associate professor in the Department of Chemistry and Chemical Biology here at Northeastern. He has a BS in chemistry from Harvey Mudd College, and then completed his PhD at UC Berkeley in the lab of Matt Francis, and then a postdoc at Boston University in the lab of Mark Grinstaff, before starting his independent independent career at Harvard. We were lucky enough to recruit him to Northeastern in 2020, and he is broadly interested in topics related to biologically inspired materials, protein engineering, self-assembly, and biointerfaces. Professor Joshi has won numerous awards, and most recently, he and his research team were selected as a grand prize winner in the National Science Foundation's 2026 Idea Machine competition, which sought grand challenges to help shape the US research agenda for years to come. The team's proposal was one of only 4 grand prize winners selected from close to 800 submissions. So without further ado, I will turn it over to Neel.
Thank you, Dean Sive and Penny, for the wonderful introduction. Thanks for the invitation to speak at this cool new seminar series. I always value the opportunity to talk to folks from a variety of different backgrounds, and in the handful of times I have been able to present to a really broad audience, sometimes people who don't necessarily have a scientific background, I've always appreciated the insightful feedback and questions that I get that often are different than what I would normally encounter at a scientific conference, for example. So I'm definitely looking forward to that. Hopefully you can all see my screen now. So I will get into it. My lab focuses on making biologically fabricated materials from engineered microbes. And I'll explain what I mean when I say that. And so what we have around us all the time, all these objects that are popping up, I'll call our built environment. These are things that we make that surround us all of the time. And a lot of these are in part or in whole derived from non-renewable resources like petroleum. And so this is partially a problem that my lab is seeking to address. And it is also a problem that is part of some of the grandest challenges facing humanity right now. Both ends of this process in terms of the resource extraction at the start of this process, and then also the pollutants and byproducts of this process are causing massive problems in ecological damage. And then also, of course, climate change is a direct byproduct of this type of approach to our materials economy, making the environment that surrounds us. And so us, and including many other people, are interested in rethinking this paradigm and figuring out how we can derive building paradigms from biology, which is able to do this, of course, in a renewable manner. And so biology is able to make massive amounts of various structures that we are kind of familiar with and even use to some extent in our own built environment. And it's able to do this in a renewable way, in a kind of a more circular economy that avoids a lot of the problems that are associated with that linear materials economy that I presented on the last slide. So even if we take the total amount of petroleum that we could use as an estimate of how much kind of raw material we have to work with, it's about 150 times 10 to the 9 tons, billion tons, that is. And if we compare that to the amount of cellulosic biomass, like plant biomass and other forms of biomass made on Earth yearly, it's about half of that. So every year we make about half of the total amount in biomass of the amount of petroleum reserves that we have. And so that it clearly points to a great potential to make things in a different way on massive scales that is more in line with the way that the Earth works in a natural way, that could help mitigate some of the biggest problems facing us right now. And so when we try and figure out how biology makes structures and how we can derive inspiration from that, I often use this as an analogy where this seed contains embedded within it all of the genetic instructions it needs to assimilate water, energy sources, and nutrients from its surroundings, and then undergo a process of hierarchical structure building. So cells embedded in this seed will make materials in the form of biopolymers and make more cells and create larger and larger structures, eventually to create this tree, which is kind of the mature form of this material. All of these processes are directed by living cells, and even after the tree as a whole is made, the cells that made the tree remain embedded in the tree, and they can contribute to dynamic properties of the tree. For example, they can move the tree, grow in certain directions, they can have some healing capacity, etc. And so we're interested in deriving design principles from how biology builds materials, but using engineered systems to build materials in a similar way. And so what we're interested in is something called engineered living materials, where we would start with a cell that we provide a set of DNA instructions that would program certain functions into the cell, including ones that are very similar to what is happening here with the seed. So the cell could proliferate, and based on the instructions that we give it, it could build specific three-dimensional shapes, eventually to produce macroscopic materials where the cells remain embedded in there and can contribute to dynamic properties. And so we think that this could be an approach to address some of those problems, and also an approach to creating materials that have fundamentally new properties that mimic biological materials, like stimulus responsive behavior, being able to respond to the environment, et cetera. And so this is kind of a blue sky goal of what we would like to accomplish in my research program. So if we consider the way that we make a lot of those things that were on the first slide, all those petroleum derived products and other similar things, it's usually through a process called top-down fabrication, which means we take a bunch of molecular components and then we, for example, would mold them into a specific shape, as is shown here. And that's how you get the wonderful shapes that make up all the plastic objects that surround you every day. And if we contrast that to the way that biology builds materials, we could call that a bottom-up approach. And so if we look at this tree, the molecular components that build this tree, like the cellulose that forms the cell wall and forms the bulk of the tree's structure, is these polymers are bundled into small bundles, which then further bundle into larger fibrils, et cetera, until we form this fully formed tree. This is certainly a common feature of the way that biology builds structures. So our tissues are built in a similar manner with different biomolecules. So in this case of this tendon, we'd be talking about collagen molecules that are similarly hierarchically bundled to create larger and larger shapes. And how is biology able to do this? It's able to do this based on the precise sequence control that it has over the polymers that it's making. And so if we take a look at this collagen fiber, this collagen polymer that builds up the tendon, and look at an individual molecule of collagen within that structure, we can see a very specific sequence of building blocks that enable it to perform this self-assembly. So these three amino acids in this case are the building blocks: hydroxyproline, glycine, and proline. This pattern is repeated over and over again. And this is what allows, in part, for this polymer to self-assemble into this very specific triple helix and then to undergo higher order assembly into the larger structures. And so we're interested in mimicking those types of bottom-up approaches to structure building using living systems. And once again, the reason that this is not really very feasible with the top-down approach and using petroleum-derived polymers, for example, is we have much less control over the sequence of building blocks that goes into those. So a polymer is one molecule that gets strung together a bunch of times to create a much longer molecule. And so each one of those units essentially has to be the same, or you could play some games by introducing other components as well, and create copolymers that have multiple components, but you don't have the same level of control over the sequence that you do with biological systems, which use the wonder of the protein transcription and translation to really precisely program a specific sequence into a protein structure, which enables the functions that I showed you on the last slide. So what we're interested, and so the outcome of this is that for polymers like this, you get a kind of very non-specific folded shape. Whereas for these very precisely sequence-defined polymers, you're able to achieve very specific shapes like I showed you on the last slide and are shown here. So this is, again, collagen that I'm showing you here. And so what we're interested in doing is leveraging the power of biology to create those very precise polymers in order to make materials. And so this is an electron micrograph. It's false colored. These things are not actually these colors. We just kind of painted them in after obtaining the image. And so this purple thing that you see here is an E. coli cell that we've engineered. And it's sitting on this green background, which is just a glass slide. And this yellow mat that it is sitting on is a bunch of protein fibers that it has made. And so the paradigm that we're interested in is being able to take renewable feedstocks and engineer this non-pathogenic E. coli strain with the capability to make specific protein-based polymers in order to make materials with custom function. Similar to the way that a polymer chemist might design polymer molecules to have specific functions that are advantageous for what we want to do in our daily lives. So you could think of like the non-stick properties of Teflon, or the water resistance of the plastic in a water bottle, or specific toughness that you would need for kind of load-bearing structures. And so our starting point for this was bacterial biofilms, which is maybe the simplest example of biological structure building that exists in nature. And so what bacterial biofilms are bacteria that are living embedded in a a polymeric matrix of their own making. And so all these cells that are living in this biofilm are secreting polymers in the form of proteins, DNA, polysaccharides, et cetera, into this space in between each of the cells. And that creates this, this slime, this structure that you are maybe familiar with from the slime that you would find on the surface of a pond, or on the back of your tongue when you brush your teeth. So a lot of people are interested in getting rid of biofilms, rightly so, because it contributes to things like bacterial persistence and infection. But if you look at the biofilm matrix from the perspective of the bacteria, it plays some useful roles that maybe we'd want to mimic. So for example, the matrix enables a resistance to harsh conditions like harsh pHs, temperatures, and solvents. So the polymers have to be robust. The cells can make more of these polymers, and so it's a self-regenerating material. It's surface adhesive. It allows the cells to stick to specific surfaces. And it also has a vast surface area that you could exploit to control interactions with their surroundings. And so this is another electron micrograph of an E. coli cell that's kind of nested in a matrix of biopolymers that it has made. And so this is a picture of what a biofilm could look like. So we're interested in engineering, especially the protein components of this matrix that the cell is embedded within. And our entry point for that is this system called the curly system that naturally occurs in E. coli. So what we do is we kind of hijack this system in order to create things that we want. And so the way the business end of this, and just to orient you here, these are two membranes of E. coli. So this is the outer membrane. So this is outside the cell at the top of the slide and inside the cell at the bottom of the slide. There's a lot of details here that I'm going to skip over, but the business end is this one protein molecule called CSGA, which is secreted from the cell into the outside, the extracellular space, and then self-assembles there into fibers. So you can see that it changes its shape, and then one protein stacks on top of the other until it assembles into a very long fiber that's much longer than the cell. And what that looks like is something like this, where these ovals are, again, the non-pathogenic E. coli that we've engineered. And all of these nanofibers, these small wispy things around the cells, are made essentially of this single protein, CSGA in this case, in this electron micrograph. And so we see this as an opportunity where we could just change a single gene or a small set of genes that encode for the formation of this molecule, and we could have a dramatic impact on this material as a whole. And indeed, that's what we did when we first started this kind of project in the lab, where we were taking, again, non-pathogenic laboratory strains of E. coli, knocking out the genes that encode for the native curly fiber machinery, the native CSGA, and complementing with extra DNA that we add into the cells in the form of plasmids that encode for an alternative version of CSGA. In this case, we add this extra green domain onto CSGA as like a pendant molecule. And so you can see that after this protein is produced, it makes something that we've schematically drawn like this, where we have the CSGA molecule shown in yellow and the extra domain that we add shown in green. This gets secreted into the extracellular space, just like I showed you before, and then it self-assembles into these fibers, except each unit along this fiber is displaying this green domain. So the green domain gets displayed all over the surface of of the material as a whole. And by choosing what the function of that green domain is, we can then influence the properties of this material as a whole. And so we've done that in various different ways and done a lot to kind of develop the basic ability to engineer the system in this way, made a bunch of different materials this way. And I'll tell you just really briefly about a few examples of what we're doing with it. And so we've taken it in a bunch of different directions. And I'll focus here on our efforts in manufacturing and also in the biomedical arena. We've also done some stuff with remediation, which I won't talk about as much. And so the core aspect of what we're able to do here starts, we want to make it as streamlined a process as possible, because this is really what we're going to need in order to manufacture large amounts of material this way. And so what we start with is bacteria that are just growing in broth culture. This is a standard way of culturing bacteria in the lab. We induce them, we program them to produce those protein fibers that I've been talking about. And then all we do to harvest our material is filter them through a filter paper. So you can think of like the same type of filter that you would use to filter coffee. It turns out that an interesting feature of these protein fibers, which I'm showing here in green, is that they're extremely robust. They're even more resistant to various different conditions than the cells and everything else that is in this mixture. And so what we can do is wash this material on the surface of the filter with a bunch of different harsh conditions, which basically get rid of and wash away all of the other components here, except for the fibers, including cells, DNA, et cetera. So all that's left on this filter surface is are the protein fibers that we ask the bacteria to produce. This is what that looks like. This is another electron micrograph. This circle here that I'm highlighting is a pore that's in the filter underneath, and all of this material, this mat on top, is composed essentially of that single protein CSGA. If you zoom into this, you can still see the characteristic nanofibrous mesh that I showed you before. And we're able to essentially get a semi-pure product out of this. So it's a good starting point for making materials in a very streamlined fashion. And we can also make materials, depending on how we do these washes, in different forms. So what I showed you before was like a dry mat that you could scrape off of the filter surface. We can also make things like hydrogels, so hydrated structures that you could extrude, that could interface with biological systems. We can also take those hydrogels and dry them into bioplastic-like structures. So if we take this gel, this is showing one of the gels that we can harvest from that filtration protocol, and then dry it in a mold, for example, we can create this nice transparent square, which is made completely from bacteria. And these are some prototypes of bioplastic-like materials that we've made in the lab. We can tune the properties of these materials to some extent, where we can make them either rigid, as is shown in this these images here, or flexible, as shown on the bottom here. And so we have a current NSF grant to investigate the use of this platform for making biodegradable bioplastics. Another example of this is this house that we made, where each panel of this house is made from one of those gels that has been dried, and then we glue the whole thing together with a few drops of water at the interface. So it's a cool way to build three-dimensional structures, and the entire house was made completely from bacteria. We didn't add any exogenous material or polymer to the structure. So it's a house made completely by bacteria. As you may have guessed, this is a slight challenge then for using this as a bioplastic. When people think of plastics, you're thinking of things like water bottles and stuff like that, where the plastic needs to come into contact with water. And these specific prototypes do not withstand water, so they'll be dispersed in water. We think there's already kind of an application for these types of materials. For example, you could think of the detergent pods that you put into your dishwasher, which have a water-soluble polymer surrounding them. And that polymer itself is not actually biodegradable, despite its ability to dissolve. And so we could replace those already with materials like the ones I'm showing here. And then the NSF grant that I mentioned, its focus is specifically on engineering this system further to create water-resistant versions of these bioplastics. So it turns out that we can also expand the range of material properties we can access by combining our gels with other materials to create composite materials made of multiple components. And so what I'm showing here is a bunch of other materials that are much more abundant than our bacterially derived material, that you might actually think of building large structures with, houses, other things, etc. And you can see that in their kind of native or pure form, they're not so easy to work with. So they're powders or gels. If we combine them with our bacterially derived gel, we can extrude them like a 3D printer into specific shapes, which is what's being shown here. And then once they dry, they create very rigid structures where these particulate things that we've isolated from these abundant sources are essentially held together by a glue made by the bacteria. And we can create relatively rigid objects that you might actually consider building larger structures out of, like load-bearing structures. So that's our efforts in manufacturing, and we have some current projects investigating their use for things like packaging materials, bioplastics, et cetera. I'll transition now to some of our efforts in the biomedical arena, where we're interested in using the same type of platform, these engineered bacteria, to treat diseases in the gut. And so bacteria already live in your gut. You can, people are generally familiar with the concept of, for example, ingesting things like probiotics. You can add bacteria to the gut. And so our interest is in using our engineering capabilities to use living bacteria to deliver therapeutics inside your GI tract. And our initial target has been in the treatment of inflammatory bowel disease, or IBD, What happens in this disease is that it's a chronic state of inflammation inside the gut. You form these inflammatory lesions, which are these white patches shown in these endoscopic images taken from inside the GI tract. And it's a huge problem for which there are not very good solutions currently. People are typically diagnosed young, and they have this chronic condition that needs to be treated usually with systemic immunosuppression, which causes other problems, susceptibility to infection, et cetera. So there's a lot of interest in ways to create local therapies inside the gut to treat these types of diseases. And so our approach to this has been to start with now a different strain of E. coli, which is already known as a probiotic. So this has a, it's called E. coli Nestle. It's sold over the counter in some countries under the brand name Mutaflor, in the same way that you can go down to CVS or another drugstore and get a pill that contains other probiotics in it. So this is similar to that. It has a strong track record of safety in humans. And what we're doing is recapitulating our engineered system where we're producing these engineered or altered curly fiber materials inside this probiotic strain. And the idea is that you would ingest this engineered bacterium would proliferate inside the GI tract and then create that material in situ. And we would design the material such that it had therapeutic function and was able to treat some gut diseases like I showed you on the last slide. And so just to kind of cut to the chase with how this works, what we do is we add this green domain is now going to be a therapeutic domain, specifically one that is anti-inflammatory and contributes to wound healing so that we could close the wounds that were in the last, those inflammatory lesions, the white patches. So this is what that system looks like. When you suffer from this disease, those inflammatory lesions essentially cause holes to form in your gut lining. Your gut is responsible for keeping certain things out and allowing other things in. And if you get a hole there, then you, basically bacteria, particulates, et cetera, cross that barrier and exacerbate inflammation. And so what our system does is our bacteria are able to exist in this at the site where these lesions happen and produce this living patch in the form of these protein polymer networks that are able to promote wound healing and close the wound. So the last thing I'm going to tell you about here is some of our recent results showing that this actually works in a living disease model in mice. This is a little bit technical, but I'll walk you through it. And so this model is one that is really common to use in investigating IBD treatments. What it consists of is feeding mice this polymer, which injures their colon in a way that mimics colitis. And then what we're doing is investigating our ability to prevent that inflammation from happening by feeding the mice our engineered bacteria. So we begin, this is a prophylactic model. We begin feeding our bacteria before we injure the mouse. And then we start the DSS feeding to cause injury at day 0. And then we'll remove the DSS at day 5 and look at recovery. And what I'm going to show you here is weight loss curves for a bunch of different mice in these cohorts. And so the blue trace here is a negative control where mice have not been injured. And you can see that they gain weight over the course of the experiment, as one would expect. The other 3 curves down here, red, green, and purple, are cases where the mouse has been injured with DSS, so it has inflammation in the colon, but they have received either no bacteria not producing any protein fibers, or bacteria producing protein fibers with no anti-inflammatory domain. And you can see that they all behave very similarly. They start to lose weight after the injury and eventually start to recover at the end here. And the orange curve here is where we're delivering our therapeutic vehicle that's producing that anti-inflammatory patch, that anti-inflammatory material in situ inside the gut. And you can see that we're ameliorating the weight loss and we're able to recover back to where the healthy mice are at the end of 10 days. And this data is mirrored by other measures like disease activity index, which is a measure of disease severity using other kind of physiological parameters. So our curly bound anti-inflammatory domains are able to ameliorate inflammation. And we're now investigating what it would take to work towards clinical translation with this idea. So I'll just close there and highlight a few examples of things that I think we compare to in favorable ways. So sometimes when I get to the end of my talks, the concept of making, excuse me, large objects, large scale materials out of bacteria or engineered microbes still seems like sci-fi. But so I just want to highlight a couple examples of people who are already doing this on relatively large scales. And so one example is shown here. It's a designer from the UK who takes bacterial cellulose. So the way she grows this is in her bathtub with a kombucha culture. So it turns out that if you do that, you can create this mat of cellulose material at the air-water interface. So she does that in her bathtub, forms these mats, and then stitches them into garments after drying them on the roof of her flat. And so she's growing clothes in her house, essentially. It's a cool approach to textile fabrication. She's got a TED Talk. Her name is Suzanne Lee. You can check it out if you're interested. In that talk, she's wearing this this cool looking vest. And she says that while it's a cool demonstration piece, it doesn't make for a very functional garment because the cellulose that it's made of will swell in humid conditions. And so if you go outside on a humid day or it's raining, you would be sopping wet. And so it'd be nice to have bacteria that are able to make materials that are better suited for the types of applications that we want to use them for. Another example are mycelium-based materials, where various companies, including the one shown here, are taking agricultural waste and putting it in a mold, inoculating that with fungus, which eats all of the cellulose that is in the twigs and stems and leaves, etc., and then creates a material to bind together all of the stuff that it can't digest. And when you do this in a mold, you can come out with specifically shaped objects like this. And their business model is to create packaging materials so that when you get this corner that I'm showing here, in your box of like a new computer monitor that you're building, that you've bought or something like that, instead of it being made out of styrofoam, which is completely non-degradable, it can be made out of this mushroom or fungus-based material that you can compost in your garden. And so this company has a fabrication plant in upstate New York that's already making this type of material on the ton scale. And so this is quite a viable way to manufacture things in large scale. And what the stuff that I showed you that my group is doing offers to this type of manufacturing paradigm is the ability to not only rely on the types of polymers that biology already makes, so things like cellulose or this mycelium-based material, but to really think about it at the molecular scale and make completely new synthetic biopolymers that have never been made by nature before, that we can program functions into and enhance the types of applications that they can be used for. I was going to mention this award that my postdocs applied for. Penny also mentioned it in her introduction, but basically it was this open-ended competition to solicit ideas for grand challenges to shape the National Science Foundation's research agenda going forward. And so these two postdocs, Anna and Avinash from my lab, did submit and win their idea based on some of the stuff that I talked about called engineered living materials. You can check out a video explaining it further on the NSF's website if you are interested. And industry and funding agencies are also starting to take notice of this phenomenon, or this paradigm of manufacturing things this way. So this is a quote from the CEO of Ginkgo Bioworks, a biotech company that is here in Boston, saying that biology is a good way to make physical goods. And then also announcements abounding from funding agencies along similar lines. So I will close there and thank all of the folks in my lab that did the work to make this possible, and these funding agencies, and you for your attention. And I'm happy to take any questions.
Neel, thank you so much. That is just brilliant work. I think, it is absolutely true that the greatest engineering that has been done anywhere on the planet has been done by living systems. And I think you're capitalizing on the engineering that living systems can do. It is far in excess of what humans can do in terms of, manufacturing in ways that don't capitulate from life. So I think, thank you for sharing that really outstanding, innovative work with us. There's some great questions in the chat, and I'd like to encourage everyone to please add your question to the chat. There is no question that is not worth asking. If you would like to ask a question, please do, absolutely. If this is not your field, no worries, just ask your question, and it will be helpful for everyone to answer it. Let's have a couple of questions here. Does the incorporation of the coli genes reduce E. coli growth, cell growth and efficiency? And, what does that mean for the manufacturing process? Do you have to pay attention to that, extending this question a little bit in getting out the materials that you're trying to?
Yes, absolutely. In fact, I definitely glossed over a lot of that in the interest of time, but we spent quite a bit of effort and are still spending effort to figure out how to make large amounts of material without impairing the growth of E. coli. In order to make this viable as a manufacturing paradigm, we need to make as much as possible because, despite the problems that we're facing, oil is still pretty cheap and it's hard to compete with. And so especially for some of the manufacturing things, maximizing the amount that we get out of E. coli is a huge challenge. And so we have done quite a bit there. I think there's still a long ways to go in terms of maximizing that, but there also, the pathway, I think, is well trod based on other things that have come before us, things like biofuels, et cetera, in terms of how to figure out how to get these microbes to make more and more of the particular molecule that you're interested in. So I think there's a clear pathway.
Outstanding. Thank you. Here's a question that I think you'll probably need to deconvolute a little bit for the answer. It's a very good question. Does E. coli nestle colonize the gut? What's stopping it from continuously producing the biofilm in the gut?
Yeah, so the way that I like to think about it, and it has been explained to me, is that you can think of your gut, especially, you can think of your gut as like a hotel where all of the rooms are microbes that are living inside of your gut. And so when you take probiotics in a pill, they typically don't stick around for very long inside your GI tract because it's like going to a hotel where all of the rooms are full. There's no place for them to reside and stay for a long period of time. And so that is also the case with Nistle, although one of the reasons we like it is that it sticks around for a little bit longer than the lactobacillus that you get in your probiotic pills. So people have done now human trials on this and characterized it, started to characterize it pretty well. And so it turns out that if you take a pill that contains Nistle, even engineered strains of Nistle, it will last about 2 days in your GI tract. And if you stop taking it after that, it will be washed out, essentially. And so we see this as an advantage. It's what we would want to do to implement our therapeutic system is to have a daily pill. That way you can kind of maintain a steady state of Nistle inside the gut and have this therapeutic function happening. If you ever encounter any problems or need to go off of the medication, you can just simply stop taking the pill and it should be gone in 48 hours. There are other efforts, especially to treat other chronic diseases. People who are interested, I would say this is at an earlier stage of research, interested in more permanent changes to the microbiome. And of course, attended with that, or what would come along with that, is an increased attention that needs to be paid to safety, biocontainment, things like that.
Very interesting indeed. I'm going down the list here, some terrific questions here. What are some of the remediation uses of the types of approaches that you've told us about today?
Yeah, so in terms of remediation, it's the main power of the platform that we have with our curly fiber engineering is the ability to make essentially like membranes that are able to isolate a single component or a small group of components from a complex mixture. So based on kind of molecular recognition, like you can think of an antibody binding to a specific antigen, it only binds to one. It can encounter many, many things, but it's able to bind specifically to one. And so we can put antibody-like things on our curly fibers and create a membrane, like I showed you on the surface of that filter membrane, that only has affinity. It's only able to bind to a single component. So this type of structure is useful for certain types of remediation, especially ones where you're trying to recover valuable things from waste streams and then reuse them, reincorporate them into other materials. So one of the papers that we published, for example, was we engineered our curly fibers to have lanthanide binding domains on them. Lanthanides are those metals at the bottom 2 rows of the periodic table that aren't typically connected when you see the periodic table. And they're really valuable for making all sorts of stuff. Your they're in windmills and your phone screens and all sorts of stuff, but they're hard to isolate. And so what we can do is create a material that we can put on a filter membrane that's only able to bind to those. So you could take a waste stream from a mine and recover all of the lanthanides from that and leave behind all the other metals that you're not interested in. So that's an example of something that we've done with this system.
Very interesting. Thank you. You talked about a hostile wash used to kill everything but the proteins so that you could get out, your material to make these really cool bioplastics, really fantastic. What happens to all the hostile wash used to kill everything but the proteins? I think the question is asking, is this some kind of a waste product that you have to be very careful with? Jerry, if you'd like to clarify your question a bit more, it's really interesting. But Neel, maybe you could comment, I think, on the production process here.
Sure. Yeah, that is how our kind of starting point for doing this did involve trying to work with just the protein material on its own. We were very interested in getting rid of everything else because we wanted to we wanted to understand the properties of the material alone. I think going forward, and we've already started doing a lot of this, some of the prototypes that I showed actually keep the cells there, meaning it doesn't try to get rid of the cells and other material. We can do these types of fabrication procedures in a way, regardless of whether the kind of material from the cell is there, in a way that maintains cell viability so the cells are alive. And then you could, for example, take a piece of that bioplastic and put it back into bacterial culture, and then it would proliferate and grow more material. Or you can do it in ways where you kill all of the cells so it's not able to do that. It's an inert material. And in terms of the wash, it is harsh in the sense that it kills all the bacteria. So that's one of the ways that we can get rid of bacteria or inactivate them, kill them. But it's not necessary, and we can kind of tune that based on what we're looking for.
Terrific. Thank you, Neel. Jerry is asking particularly if the wash is a pollutant that you have to consider in your production.
The wash doesn't have any toxic things in it. Mm-hmm. And like I said, we do think it's inefficient at the very least, because you're losing a lot of the mass. The stuff that comes out of the bacterial culture is about 50% by mass, the protein, and 50%, just cells that made the protein. And so we're interested in keeping that material of the cells themselves around so that we can increase the efficiency of the process overall, which involves even less kind of washes and purification.
Oh, yeah. Terrific. Now, so here's an interesting question. If you're using E. coli as a medication in a way, do you get infection from it? So we think about, E. coli as something that can cause disease, and here you're using it as a protectant. And indeed, we all have some E. coli in our guts already. How do you weigh up the potentially deleterious effect of using bacteria as a medication compared to having bacteria causing a deleterious infection?
There's definitely a concern there in that it's fundamentally different than other drugs that we are used to taking, right? Where you take a certain amount and then that amount is going to decrease in your body as it gets excreted. And so with bacteria, it's a living therapeutic. It can make more of itself inside the gut. So you do want to be careful about having kind of runaway problems or having the bacterium exist in places where it shouldn't be. Luckily, the gut is pretty tolerant to having things like other bacteria proliferate there. And we're using a strain to begin with that has a pretty strong track record of safety in humans, including for the treatment of IBD specifically. And so, we're relying pretty heavily on that as a starting point to do this. Just the fact that through experience and trial and error, people know that this strain doesn't interfere. It also doesn't have any virulence factors, which are genes that people know are associated with pathogenic or bad functions inside the GI tract. So that's been our strategy so far.
That's very interesting and very careful in terms of considering how to use these as novel therapeutics. Absolutely. Here's a nuts and bolts question. If you grow E. coli in a shaker flask, do the cells coagulate? Do they stay separate from one another? And let me extend that to ask how this impacts the manufacturing process.
Yeah, so we actually grow them in such a way that we promote their aggregation. So the formation of those fibers, if we do it under certain conditions, can cause the bacteria to cluster and aggregate into larger structures, which makes them easier to harvest by filtration. So we can use filters with larger pore sizes in order to facilitate that. And so we try to rely on that to enhance our ability to harvest material. And also, we can even further enhance that. I didn't show this data, but we can create two forms of curly fibers in the same pot with a binding interaction. So we can create one curly fiber variant that is programmed to bind to another one, and then we can create bundles of fibers and start to build up larger and larger structures that way. So it's a process that we're looking to exploit to build larger structures.
Very interesting. I have a question that I'm going to interject because I see a moment to do it. You know, when we think about the plastics industry, it's a chemical industry. As I said right at the beginning, it's one of these great chemical industries built across the world. But you have a new plastics industry. You have a bioplastics industry that has this, you know, wonderful property of biodegradability and not having the sort of sense of dangerous unknown chemicals to begin the process and the pollution and the byproducts and the side effects associated with your products. How, though, can you think about scaling this so that you're at the level of the global plastics industry, let us say? If you, so there's I have, you know, there are multi parts to this. What kind of scale could you get from your process? And then I suppose there's a corollary to ask, what kinds of existing plastics could you replace with these bioplastics?
Yeah, so I mean, there are bioplastics that are already made on quite large scales. So I think existing biodegradable bioplastics account for not very much yet, but about 1% of the total plastics industry. And so you may have encountered things like compostable utensils in a cafeteria or something like that. And so those are typically made from either starch-based bioplastics, so it's directly derived from plant material, or from other polyesters, some of which are made by microbes. And so people are investigating those and expanding their kind of reach or the slice of the pie that they account for. But one of the things that hinders that is that you can't, with these limited set of biopolymers that have these properties of being compatible with manufacturing techniques and also being biodegradable, you can't access the range of material properties that you can with conventional petroleum-derived plastics. And so that's in part what we are trying to address, and other people have other approaches as well. And in terms of how to think about scaling, there's, I think, maybe a couple different ways to think about it. One would be more centrally, like a central processing or manufacturing unit. So you could have large manufacturing plants that have huge vats of microbes churning this stuff out. And then another way that people are also interested in involves a distributed manufacturing paradigm, where you could have a small reactor on a farm that is able to convert waste materials into valuable materials that you could either use and fabricate stuff out on site, or just sell to another company that could process it into other things. And I think it will have to work together with the existing plastics industry. It's not like we can just transfer over completely wholesale all at once and kind of solve our problem. First of all, there's already a ton, actually many tons of conventional plastics out there. And so figuring out what to do with that is a huge problem. Some of our really early stage efforts right now in the lab are seeing if we can make composites of like microplastics that are already in the environment with our biologically derived fibers to kind of reincorporate some of these waste products into the material economy. But yeah, I think we'll have to work together.
Outstanding. Let me ask if there are other questions that you'd like to put into the chat, or if someone would like to say a question out loud, that is just fine also.
I ask a different question about scale. Is there a limit to what you can append to the CSGA gene?
Yeah, it's difficult to like predict a priori how the fusions will affect the efficiency of the process, but definitely kind of anecdotally, larger domains are harder for the cells to produce. They can still make pretty large things. Like I think our record is 288 amino acids on top of the curly CSGA sequence. So that's actually larger than the monomer unit itself. And we can still make it. The cells grow a lot slower, so we have to put more effort into the metabolic engineering to counter that. Yeah, we can make like fully folded protein domains, not just peptides there. But then we do have to think about taking a hit on how much we can produce per cell.
Okay, thank you. Thank you. Diana has a question that is somewhere from a very interesting question. Have you thought about using kombucha as a culture for your biomaterials?
Yeah, we haven't used kombucha. Other people have. We have a similar process where we're interested in these composite materials. Like the curly fibers that I talked to you about are really great in terms of their customizability, but they aren't as good as other materials in terms of their scalability. It's harder to produce massive amounts of them. And so I think the way forward for, depending on the application we're interested in, is to have materials like cellulose that bacteria and plants already know how to make massive amounts of these materials, and have our fibers act as a kind of additive that can enhance the properties of those existing materials. And so we've done that, and we have actually a preprint out that we just put out a couple of days ago showing that we can have a two bacteria system and make capsules that are made of E. coli. So we can make little capsules about 1 centimeter in diameter, where they're made of first the bacteria producing, or the cellulose producing bacteria make the capsule itself, and then our E. coli will add to that and add a curly fiber shell inside of that. And that's similar to a process that other people have done with yeast that are derived from kombucha as well. So I think that's another frontier of this type of manufacturing, is having multi-organismal systems that are kind of cooperating to build a material that's bigger, that's better than the sum of its parts.
Thank you. Thank you so much, Neel. I want to thank everyone for attending today. I want to thank Neel for sharing really his extraordinary research. This is at the frontier. It's at the frontier of chemistry, of protecting the environment, of promoting human health. This is very important, extremely important work, as indicated by the accolades that Professor Neel Joshi's research group and himself have already gathered. We so appreciate your support. Research is incredibly expensive. Supporting our student trainees is such a challenge. It is something that we are committed to, but it is always a challenge. And we so appreciate the support, your involvement in our programs and in the trajectories of investigators such as Neel. Our chemistry and chemical biology department is outstanding, and the research that is done there is absolutely at the top of world class. I started by telling you how committed we are to giving undergraduates research opportunities. We are committed to expanding our graduate PhD experiences. We are committed to expanding our research efforts in the broadest sense, and so to promote the excellence of the College of Science and our wonderful Northeastern University. I encourage you to speak with us about your interests in connecting with the College of Science, with Professor Joshi, particularly with chemistry and chemical biology, and with other aspects of the college. I see someone who has joined us, and I'm delighted just at this last moment to welcome Kevin Thompson, who is our new Associate Dean for Research. Welcome, Kevin. It's such a pleasure to have you here. Those of you who know us will have seen the announcement that our former Associate Dean for Development, James Poulos, has moved into our Central Advancement Office, but still a wonderful collaborator for our college. And we're so delighted to welcome you, Kevin, to our Associate Dean for Development role.
Thanks so much. So glad to be here.
Wonderful. And you will all be knowing Kevin going forward. It's a great pleasure to have him here, just in Boston, having driven, excuse me telling everyone, Kevin, having driven 12 hours straight from North Carolina to get here. Wow, that is something very, very impressive there, and plunging right into your role here. Thank you so much. Such a pleasure. So in closing, thank you so much, Professor Joshi, for sharing your outstanding research with us. Thank you, Professor Buning, for being the introducer and for being here and giving us your valuable time. Thank you all for sharing your time with us. We look forward to seeing you at the next College of Science Connects seminar, and we look forward to connecting with you in many ways. Thank you so much.